In previous lessons, we've learned a little bit about the basics of how cells operate. In this lesson, we will focus our attention on a single-celled organism called amoeba. They live in watery environments all over the world, and they are hunters, so they cannot do photosynthesis. This means they must eat. In this picture here, we see amoeba stained with various different colors of stain. You can see they're single-celled. The more darkly stained object would be the nucleus. Some amoeba actually have multiple nuclei, which is interesting, but lots of others have just one nucleus, as is typical in animal and plant cells. Here we see the amoeba and its method of execution. It is going to extend portions of its membrane around its victim, enclosing it, trapping it, and that will become a food vacuole where digestion will take place. Now, as a single-celled organism, of course, the amoeba as a cell is filled with cytoplasm, and there are all kinds of proteins doing different jobs in the amoeba. We'll introduce a different kind of metaphor here. We can think of proteins as the workers of the cell. So if we think of a cell as a city, then the proteins would be the workers. Now, in a city, we might have people working at the power plant or in a grocery store or taking care of garbage or fixing the sewer lines or growing food, etc. For a cell, proteins are doing all the jobs that need to be done so that the cell can remain alive. Now, proteins are molecular adaptations, and that means that they are parts with purpose. And for biology, parts with purpose have an evolutionary history, so they arose by natural selection. This is the process that Mr. Darwin alerted us to, and we'll study more about this later in the course. So as adaptations, proteins have an evolutionary history. So if we think about the carrier protein here in the membrane of a cell, or some protein that breaks down food, or maybe it attacks glucose or whatever. Here's a protein that is making a chemical bond between two molecules. These proteins have unique functions. They have unique jobs. And we can ask the question, well, how did they get their jobs? And the answer to that is going to be Darwin's process of natural selection. So we'll say more about that later in the course. But cells today have functional proteins because those very proteins helped their ancestors survive and reproduce. So every kind of protein had a first usage by some ancestor cell in the past. If it helped the cell, its DNA recipe was more likely to be passed on to offspring during reproduction. So in a moment, we're going to go through amoeba and just take a look at sort of some of the proteins and how they do their jobs. But each of these proteins has unique functions, and the proteins got their jobs by this historical process of natural selection. In a previous lesson, or another lesson, we learned a bit about how amoeba can track prey and move towards prey. So I'll refer you to that lesson. Just very quickly, here we have an amoeba here, and here's its prey, the paramecium. Both are single-celled organisms. Here the amoeba is extending pseudopods to sort of engulf its prey, and when the pseudopods join here, they will form a food vacuole. Now, in that other lesson, we mentioned 3 critical proteins, now, and that's a simplification here, but the idea is that when proteins interact in the proper way, it can generate adaptive behavior. So we have microtubule proteins, we have chopper proteins, and then we have the membrane receptor proteins. And when all these proteins do their job, that's going to move the pseudopod towards prey, and that's clearly an adaptive behavior. So with amoeba, let's pick up the story then, where the prey is in the food vacuole. So here we see a picture. Here's the paramecium in the food vacuole. Now that's the chamber that forms after the prey has been fully surrounded. So the food vacuole is surrounded by a membrane. So this is a membrane-bound structure. The membrane of the food vacuole would have been the outer membrane of the cell. But at a basic level, why is amoeba doing this? Why is the amoeba going to destroy the paramecium? Because the amoeba is going to turn this living paramecium into a pile of useful nutrients and energy, right? So the amoeba is going to turn the paramecium into amoeba parts. And so this is why cells eat other cells. Again, the amoeba cannot do photosynthesis, so they must survive in this fashion. Now, the digestion occurs in the food vacuole, so we'll in a moment we'll see that there will be digestive proteins that will begin to break down the food, and then the idea is that the paramecium will be turned into a whole bunch of useful nutrients, some of which would be energy molecules like glucose, and some of which would be simply building materials for other important molecules in the cell. So there would be amino acids to build proteins, there would be DNA, the building blocks of DNA to build the amoeba's DNA, etc. There would be lipids, fats and lipids, so that the amoeba can build its own membrane. In the picture here, we see two food vacuoles. In this one, there are two victims. Here we see another food vacuole, one victim, and here's another food vacuole over here. In fact, you can begin to see, just barely see, sort of some perished paramecia here that have already died in the food vacuole. So digestive proteins are like molecular scissors, and they're going to be creating a bunch of useful nutrients from the prey. So here we have our little blue Pac-Man and women proteins chopping up the paramecium into a pile of nutrients. Now you recall that large molecules cannot cross membranes. So just as we said on the outer membrane there are channel proteins, also on the food vacuole membrane there must be, we'll call them transport proteins. So the transport proteins are going to shuttle the nutrients from the food vacuole out into the cytoplasm. And remember, proteins tend to have very specific jobs, so if there are different types of nutrients, there's likely to be a different kind of protein that is specialized to transport it across the membrane. Now, one of those nutrients would have been our friend glucose here, and of course we've learned that glucose is an energy molecule, and we can think of it as storing potential energy in the chemical bonds. And to extract that energy, to get that energy, the bonds must be broken. Well, how do you do that? How do you harness the energy of glucose? You got to have a team of proteins. So we're going to have proteins that will begin to perform chemical reactions on the glucose molecule. And again, because the breakdown of glucose takes several chemical steps, there's going to be a specialized protein for each step. So here's a protein that maybe takes the first bite, and then there's another floating around ready to take the second. And in fact, you'll have a whole team of proteins ready to metabolize glucose. The resulting food molecules, so when glucose gets broken down a bit, those molecules enter the mitochondria. We learned this was a membrane-bound structure that completes the metabolism of glucose. Oxygen is needed for this chemistry, waste molecules are produced, and ATP, energy molecules, are the whole point of doing this metabolism. ATP molecules have usable quantities of energy, whereas glucose has a lot of energy, but lots of chemical reactions, as we've learned before, require smaller bits of energy. So to make this chemical bond, you only need $1 worth of energy, not the $20 in glucose. Now these numbers are a little bit made up, but it gets the idea across. Mitochondria are the metabolic engines of the cell. So again, the idea here is you're taking a fuel and converting it into another type of molecule that stores energy, but just more usable quantities of energy. And just as an engine has a fuel, there's potential energy in gasoline, and that energy will be transformed into the energy of motion, kinetic energy. So mitochondria are taking the energy stored in glucose and doing chemistry, but then repackaging that energy in the chemical bonds of ATP molecules. Now we said that process requires oxygen. Well, amoeba live in watery environments, and there is dissolved oxygen in the water. This is molecular oxygen that can go right through the membrane. Oxygen is small enough as a molecule, can pass right through the membrane and enter mitochondria. And likewise, carbon dioxide waste that's generated from the chemistry here can simply move out into the cytoplasm and then out of the cell into the environment. Now, there's a very important structure that cells like amoeba have, and it's called a contractile vacuole. And this is an adaptation for a particular problem that an amoeba will face. When they live in fresh water, the concentration of water inside the cell is actually less than outside the cell, because after all, there are a whole bunch of other molecules here. There are proteins and other kinds of molecules in the cytoplasm. So in fact, there's more water in a sense. The concentration of water is higher outside the cell than in. Now, water is a small molecule, so it can pass through the cell membrane. And so what tends to happen is that over time, water tends to diffuse. It leaks into the cell. Now, diffusion is this movement of molecules from high concentrated areas to areas of lower concentration. And so water will leak into the cell. Now, that's life-threatening, because if the amoeba has no way to get rid of that excess water, it can burst. So the amoeba has a problem living in fresh water, and it has evolved a structure called a contractile vacuole to pump this excess water and other liquid waste out of the cell. In this picture here, we see two contractile vacuoles, actually. This one here is in the kind of, it's in the open position, so it has been filling up with liquid from the cytoplasm. And now if you can see in this picture, it's a little bit smaller, and then in the picture right after that moment, you don't see it anymore, because what has happened is that this contractile vacuole has contracted. It has squeezed the contents of the vacuole out into the environment. And the way we'll understand this is that the contractile vacuole as a chamber, it's a three-dimensional chamber, but it's surrounded by important proteins. And these proteins can tug on each other, and when they pull on each other, it diminishes the diameter of the opening to the external world, of the whole chamber. And then because there is an opening to the outside world, whatever was in the vacuole gets forced out. So the key again here are certain kinds of proteins that can interact to generate force. It's a bit like the sweatpants that have the drawstring. You know, you pull on the drawstring and then the waist gets smaller. We've got a set of proteins surrounding this chamber, and when they interact, they squeeze the chamber to become a smaller volume, and that forces the fluid out of whatever opening there is, and that opening is into the outside world. Now, what kinds of proteins can do that kind of thing? Well, these two kinds of proteins, myosin and actin, are actually found in muscle cells of animals like us. They can be found in lots of other organisms as well. But this is the kind of interaction that can generate a force. The myosin protein has these little barbs that can make contact with actin. Both of these are sort of rod-like proteins, but the myosin has these little barbs. And the interesting thing is these little barbs can flex. They can generate a little force. And when all the little barbs pull on actin, that moves actin. And so the ones over here on the right are going to move left, and the ones on the left are going to move right. Now in a muscle cell, that's going to shorten the muscle cell, and that's what we call contraction. Well, likewise, surrounding the contractile vacuole on amoeba would be proteins that can generate force. Now, many important molecules in the cell are chain molecules. So here we see a chain molecule, and they can be built from units, from nutrients in food. So if this were a protein, then the relevant nutrients would be amino acids. If this were a DNA molecule, then it would be the building blocks of DNA. So the idea is that the molecules that make up the nutrients of food, so the paramecium molecules, are going to be used to build amoeba molecules. So there were paramecia proteins. They will be broken down to amino acids, and then the amoeba is going to use the amino acids to build amoeba proteins. There was paramecium DNA. It will be broken down into basic building blocks, and then the amoeba will build amoeba DNA. And likewise for lipids. Now, like other cells, the amoeba can build its own proteins because it has a recipe to build those proteins, and that recipe is stored in the molecule of DNA found in the nucleus of the amoeba. So DNA is a very long molecule. It's one of those chain molecules, and we'll learn that it can store the recipes for building proteins. So each of the proteins we've encountered would have its own unique recipe in the DNA stored within the nucleus. So whether it's a microtubule protein, or the choppers, or the membrane receptor proteins, or the digestive proteins working on food here, the transport proteins, or the proteins that attack glucose, or the proteins that generate force for the contractile vacuole, or the proteins that build important cell molecules, all of those proteins have recipes in DNA. Because DNA has the three properties listed here, it is a hereditary molecule. So to be a hereditary molecule, it has to store information for making relevant cell parts, in this case proteins. It has to be able to be duplicated so that reproduction can happen, and the information to make these proteins must be able to change over time. Otherwise, there could never be the evolution of new types of organisms, new proteins, and therefore new organisms. For amoeba, reproduction is simply a matter of then duplicating DNA and then splitting into daughter cells. But DNA is the critical molecule involved. It stores the information to make all the amoeba proteins. It can be duplicated so that amoeba can reproduce. But when that DNA is duplicated, sometimes changes happen, and this means that the offspring may be unique. There could be some differences, and that, Mr. Darwin will argue, is critical for the evolutionary process. It means that one or the other individual might have an advantage in the struggle for existence. And the one that has an advantage, if they have an advantage because of some difference in a recipe in DNA that makes a better protein, then they will pass on that advantage to their offspring. And that's the process called natural selection. We'll learn a lot more about it later in the course. So this completes our quick survey of amoeba. Amoeba are single-celled organisms, and they are living organisms. They are alive because the amoeba has a whole bunch of different kinds of proteins performing all kinds of important jobs that keep the amoeba alive.